All right, you guys, big day today as I am at Colossal Biosciences lab to learn about how lead scientist Sara is bringing the thylacine back from extinction. Absolutely insane. Let's get right into it.
All right, so welcome to the Thylacine lab. This is also where we have the dodo and mega mammals team.
Where are they, Sara? I don't see them anywhere.
We're working on it. So right now they're at this cellular level. For the thylacine, we're using the dunart as our model species.
Right.
It's the closest living relative.
Like probably a lot of the people that watch this or hear about the work that you're doing at Colossal, it's like, okay, how are you gonna make a marsupial wolf, this big, well not big, but relatively big, very cool predator
From a marsupial mouse.
out of this mouse? Like, help me out here, because
Yeah.
I don't understand it, to be honest. I get it, they're all born the same size, but how long can that surrogacy last? More importantly, how are you actually gonna make that little tiny grain of rice critter? Like, how are we doing this?
Absolutely. So when we're talking about de-extinction, there's really two big problems that we're trying to solve. One of them is the reproductive part of things. So that's where we have the surrogate, establishing the species. The second is the genetic engineering.
Right.
You have dead tissue, it's hard to get DNA out of.
Yeah.
So for whatever reason, there was this brilliant museum curator in the early 1900s who decided to place a couple of thylacine pouch youngs, or pups, in ethanol. And so usually when you have museum samples, they're in formaldehyde and other harsh chemicals.
Right, which kills all the cells.
It preserves the tissue really beautifully, but it doesn't preserve the DNA. So we can't do a cell line, but the ethanol did preserve the DNA so that we were able to do a high quality reference genome for the thylacine. So then on the other side of things, we had the stem cells. At Colossal, we have established dunart iPSC cells. Why this is so impactful for both the thylacine as well as conservation is if you have stem cells, you can make gametes. So this is oocytes or sperm, egg or sperm, from stem cells. So you can think of a species like the northern white rhino, which we can't get any more sperm or all these other things for. But if we can derive stem cells from these lines, then we can create new eggs, new sperm, and they're grow embryos full of new genetic diversity.
Interesting. Okay, you ready for the toughest question of all?
I'm so ready.
Who's gonna get one first? You, or am I gonna go find one first? Yeah, probably. But when you do, you're gonna call me right away, right? Matt and Ben have promised me, and I'm taking the same promise from you right here in front of everybody watching. I get to be one of the first people to see it, yes?
Yes.
All right, that was for me. That wasn't for anybody else. Okay. Sara, this is amazing. And I'm the biggest supporter of Colossal. I believe so much in the mission.
Yes, it's been so fun. We love having you here for us.
All right, thank you, Sara.
All right,